Is it fine?
Yes, sir. Yes, sir.
Okay. Okay, so today we discussed details about the protein-protein interactions and its therapeutic application. As you know, the proteins is a signature molecule for the identification or determination of the physiology in terms of the maintenance of the homeostasis as well as establishment of the infectious disease. So initially, we know details about the proteins and then go for the regulatory natures of the proteins, how we can make a stable proteins into the in vitro conditions, and then go for the protein-protein interactions. What is the instrumentation and laboratory techniques for the establishment of the protein-protein interactions? And last part of the discussions will be the therapeutic applications. Therapeutic application means how we identify the target proteins molecules and go for the peptide biology, and further we synthesize the small molecules on the basis of homology modeling of the peptides and use for the screening process through the in vitro technology. So now proteins, as you know that the proteins made up of the amino acids. The amino acids has the carboxyl groups as well as the amino groups. So this one is a long chain of the amino acids. All amino acids are linked through the peptide bonds, and through the stabilization into the 2D structures, 3D tertiary structures, and quaternary structures, the proteins is in stable forms or in functional form. So most of the globular proteins are in tertiary structures, but few of the proteins present into the humans that retain the quaternary structures for the functional significance. Now, stabilizing factors for the proteins, as you know that the protein is a very sensitive molecules. So during the laboratory techniques, we use the different of the buffers for the stability of the proteins. Some of the proteins are the sensitive to the temperatures. So we need to balance the temperature according to the stability of the proteins or types of amino acid linked into the primary structures. Some of the proteins are stable into the phosphate buffers, tris buffers, hipase buffers, or amino buffers. So we need to fix the types of buffers, temperature situations according to the compatibility of the proteins as well as the instrumental setup for the interaction analysis. I have taken one examples of the digestive systems, how pH regulate the protein functionality inside the cells. Suppose for the in the digestive system, some of the proteins that is pepsins, that is component of the gastric juice. As you know that the gastric pH is highly acidic, near about the 2. And when the food materials after the stomach reach into the duodenum parts, at that times, the enterokinase enzymes that sensitize the chymotrypsins and then synthesize the synthesis of the trypsins that require the basic pH. So first step is neutralization of the pepsins activity and then activation of the trypsin enzymes that require the basic pH for the activity. So this one is sensitivity of the proteins molecules inside the cells, and nature's just define the cellular systems in coordinate systematic actions. So one times it neutralizes the acidity or acidic activity through the secretions of the SCM hydrochloric, and second times it maintains the basicity by secretion of the bicarbonate ions. So through this process, it maintains the stability of proteins for the significance activity inside the cells. So now we know the buffer situations, pH sensitivity, temperatures, and ionic strength for its stability. Here the PBS solutions that shows the stability of the hemoglobins, HSV proteins, BM, and other proteins molecules in the tertiary structures as well as the quaternary structures. Now, protein interactions is important. As you know that the proteins molecules that play a wide range into of function inside the cells, and every cellular process requires proteins specifically recognitions at a multitude of the different interacting partners. That interacting partners may be the DNA, maybe the RNA, maybe the proteins, or maybe the cofactors. That's why the interactions, protein interaction is important for the therapeutic application. We can design the some of the therapeutic molecules either by mimicking the domain interaction by synthesis of the peptides molecules, or we synthesize the chemicals molecules or small molecules that can interfere the protein interactions, and we can balance the activity of the cells. So protein-protein interaction means physical interaction or physical contact between the stabilized structures of two or more than two proteins as a result of the biochemical events. So how this one is important for the researchers, mainly researchers in the structural biology, molecular biology, and drug discovery fields that report the protein-protein interaction in the context of the therapeutics, and try to identify the potential target molecules through the biophysical techniques, and then try to find how proteins interact to each others within the cellular pathway to transduce the signal from the extracellular environments into the intracellular environments, and then develop the novel small molecules that can stabilize or destabilize the interaction to treat the disease. So this one is the importance of the protein interaction into the physiology as well as the balancing of the cells. So types of the interactions, initially we need to know what is the types of the interaction, how it stabilize the physiological activity. So protein interactions may be the different, maybe happens between the different domain of the same proteins or maybe the different proteins. So on the basis of these events, we can call the stable and transient activity. Like the enzyme substrate interactions, as we know that when the two specific substrates come in contact with the enzymes that form the product, after formation of product, when the product is forms, the enzymes again enter into the inactive stage and wait for the next lot of the activity. Some of the proteins are highly stable, mainly the fibrous protein or structural proteins are stable proteins that are stable throughout the physiological cycles. We can also call the proteins on the basis of activity, that is obligates and non-obligates in natures. Some of the proteins may interact for its functional activity, and others proteins may interact within the specific conditions inside the cells. As you know that the different domains is present inside the cells, some of the proteins show the functional activity through the post-translational modification. As you know that the protein, suppose the proteins has the 2 and 3 domains. Upper domains cause the autophosphorylation event that helps in the transduction of the cellular mechanism that activate the biological reactions and amplify the biological signal inside the cells. On the basis of the interacting partners, we can classify the homotypic and heterotypic natures if identical proteins involved into the interactions, that is homotypic interactions. If different type of proteins involved into the interaction, that is known as heterotypic interactions. we can use the serine threonine protein kinase that contains the two types of the kinase activity. First one, this type of the proteins activate itself through the autophosphorylation process. Once the kinase molecules activated that cause the autophosphorylation of the one terminal ends and that activate the additional terminal end for the biological activity. So that is a homotypic type of the cell interaction because the same proteins involves same domains involved into the protein for its activity, and different types of the interacting partner present into the interactions. We call that is a heterotypic interactions. We have taken the three types of the domains. This one B parts is a functional domain. Suppose ki oxygenase domain, reducing domain, and different types of the biological activity encoded by the domain B. And this one is just A and C is a proteins that contain the two different domains, that is domain A and domain C. So on the basis of 3D structures, it has the two different loops. First one is between the A and C, that is lower bottoms, and second one is in upper bottoms. So B has the compatibility with the upper domains. Sometimes the A and C domains opens in the natural configurations or configuration of the protein sequence that reorient the A and C domains. Second, through the autophosphorylation events. If B domains show the autophosphorylations that reorient the A and C domains, and that is responsible for transduction of the signals from the extracellular and intracellular environments, and that shows the response according to the driven signals into the cells. So this one is a structural compatibility for the protein interactions. Today talk, we can discuss the different types of the proteins, that is ligands molecule and substrate molecules. Suppose A and C is a one molecules, either we keep as a ligands and B domains we keep as a analytes. So how these proteins interact and how we can analyze by the biological tools and technique. What is the conventional technique? What is the applied techniques for this analysis? And how these interactions we can use for the therapeutic applications or designing of the small molecules or drug molecules of the cells. So this one is autophosphorylation, example of the autophosphorylation domains. This one is structural proteins that retain the 3D activity. For its functional activity, requires the two factors, that is the calcium plus sign. In presence of the CAM, single molecules that cause the autophosphorylation of the one protein's molecules, and we can observe the structural component of the protein after phosphorylation has been changed. Now, second proteins that has 0% of the autophosphorylation, so no activity in the second parts of the proteins. When we provide the excess amount of CAM in presence of the calcium ion, both domains are activated and structural or topological configuration of the proteins will change, and that is functional proteins into the physiology that is responsible for the transduction of the information from the extracellular to intracellular environments. So this is a significance. This is a type of the post-translational modification for the activations, for the stabilization, and that can be helpful for the identification or for the protein-protein interactions. So what is the instrumentals or laboratory method for demonstration of the protein-protein interactions? First one is in vitro methods. We can demonstrate the protein-protein interactions in the cell-free system. Second one is in vivo methods. Within the live organism, we can demonstrate the protein-protein interactions either using the confocal microscopy or in silico approach, computational approach, we can identify the protein-protein interactions. So basically, we focus here the in vitro technique for the protein interactions. What is the different laboratory techniques available to decode the protein interaction inside the cells, and then go for the in vivo and in silico analysis. But in vitro analysis we can discuss in details. So the conventional approach for the in vitro analysis of the protein interactions is co-immunoprecipitation. Co-immunoprecipitation means when proteins A and proteins B come close to each other, that show the precipitation forms, or that acts as a acts as a mainly pull down of the interest of the proteins according to the interacting proteins. So we can know details by the topological configurations. This one is the cells. Here the proteins structures stabilize within the nucleus. So what is the first approach? We can disintegrate the cells. We can extract the nuclear content. We can extract the proteins into the suitable pH. We maintain the pH as well as the temperatures or ionic exchange into the cells for the stability of the proteins. Then we can add the antibody specific to the proteins. So the specific proteins bind with the interacting partners. Then we can add the bead molecules that is specific for the antibody. So there is a complex formations. First one, the target proteins bind with the antibody. Second, the bead molecules go and bind with the Fc molecules. As you know that the antigens bind with the Fab regions of the antibody and Fab regions of the antibody, and then we bind with the Fc regions of the antibody. So that will becomes the heavier molecules, and we can easily precipitate the protein of interest through the specific elution methods. So what happens? We can wash the proteins. All of the unbound proteins are easily washed out from the cells, and then we can identify the target proteins molecules through the immunoblotting process. So this one is 2 and 4. We have added the proteins molecules, specific proteins molecules. We can immunoblot the complex molecules using the antibody directed to the second of the proteins. So this one is immunoprecipitation methods. We can add the specific antibody against the one proteins, and these one proteins interact with the second proteins. Again, at the time of the immunoblotting process, we can use the secondary antibodies that are highly specific to the secondary proteins. Means the first antibody bind with the first proteins according to the specificity, and we can identify the precipitated bond using the antibody specific to the second protein. So if both proteins interact to each others, we can identify the specific protein band through the immunoblotting process. This one is reflected in the 2 and 4. The two proteins are appears in the same positions, means ki protein 1 and 2 are interact to each other. So this one is co-immunoprecipitation methods. Means co-immunoprecipitation methods means protein 1 and protein 2 interact to each other. By using the antibody specific to protein 1, we can purify the second proteins, and we can immunoblot and detect the second proteins using the specific antibody into the column. Now second one is pull down assay. Pull down assay means ki one proteins acts as a bait and second proteins acts as a prey. So pull down assay, as you know that ki during the protein purification process, we can tag the different of the bead molecules, either the GST, his, biotin tagging to the proteins. Some of the proteins are stable through the his tagging methods. some of the protein established by the GST tagging methods. All of the tagging process is not suitable to the all proteins, so we need to try first by the HIS, then GST, and then bioteins according to the conformational stability of the proteins, and then go for the pull down assay. So first, this one is affinity ligands. These affinity ligands allow to bind with the bait proteins and then incubated with the prey proteins. All of the complex forms with the affinity ligands. Again, we wash out the unbound proteins, further elute the complex molecules, and then use for the immunoblotting, and we can easily detect the bait and prey molecules through the immunoblotting process. So this one is pull down assay. We can easily identify the proteins using the one of the bait affinity ligands and purify the prey proteins for the interactions. So this one is conventional methods that frequently used into the laboratory for the initial screening of the protein interaction inside the cells. Now currently, as we know, the science needs more confirmatory results. That's why we need to titrate the kinetics parameters. Kinetics parameter means ki what is the kinetic constant at the time of the association, what is the kinetic parameter at the time of dissociations, and if two proteins interact to each other, what will be the binding energy between the two molecules. So these methods, biophysical approach, can be helpful for designing of the inhibitory molecules or small molecules for the kinetic analysis or for the interactions minimization steps. So this one is basic sensogram for the in the SPRs. So how SPRs works? Initially, it based on the sensor domains that actually based on the prism on the basis of change in the refractive index. So this one is sensor chips present on the instrumental setups. The company fellow provide the different type of the chips based on the type of interactions and which type of the ligands and analyte used for the analysis. This one is analyze substrate analytes molecules used for the second partner for the interaction studies. Some of the analytes are in free form, some of the analytes are in club forms. The club forms indicates means higher concentration of analyte present into the buffers or inappropriate ionic strengths of the buffers that are not suitable for the homogeneous suspension of the analytes. So we need to check the homogeneous suspension of the analytes into the suitable buffers, maybe the HAPs, trees, or phosphate buffers, and then use for the interaction analysis. So when we apply the analytes into the system on the basis of the physiological, physiochemical reactions that leads to change in the refractive index, and all of the interactions appears in the three forms in the form of the sensograms. So first, this one is association phase. At the time of interaction of the ligands and analytes, both molecules are associated to each others that reach up to the saturation stage and then again enter into the dissociation phase. Means the separation of analyte from the ligands molecules. So this one is what? This one is association phase start from the 0 points and reach up to the some extents. Some of the molecules are easily dissociated from the washing steps. So this one is binding energy of the molecules. We can subtract the dissociation, that is a KT value, from the association value, that is a Ka value, and we can easily identify the binding energy. And this binding energy used for the calculation of affinity of the biomolecules. Now, if we synthesize the small molecules that has ability to minimize the interaction between the analyte and ligands, that reduce the binding energy, means ki reduce the energy between the dissociation and association phase. So we can easily know here when we immobilize the ligands over the surface sensor chips. This one is baseline value. When we applied the analytes, it bond with the receptors ligands surface, and then equilibrium phase further into the dissociation phase. So this one is binding energy from these points, these points. So this, after the instrumentation analysis, we can calculate the mathematical value or numerical value of the association phase or dissociation phase or binding energy or equilibrium constants. What will be the equilibrium constant of the biomolecules at the time of the interactions? And that equilibrium constant is used for the drug designing and therapeutic applications. We can easily define if this one is a stable interaction between the two molecules, this one is unstable interaction between the two molecules. If the interaction is pH dependent, we can change the value instrumental parameters, and we can easily say these proteins are stable during the acidic phase or unstable during the alkaline phase. We can change the salt condition. We can easily say that these proteins molecules are interact to each other as the physiological conditions. But when we increase the salt concentration, these two molecules are unable to interact to each other. So this one is biophysical approach to identify the interaction between the two molecules. Now this one is instrumental setups. We can analyze the binding kinetics through the SPR instrument. We can drug competitions, fragment screenings, irreversible binding, and protein aggregations. All the biological parameters we can identify through the SPR instrument. So this one is very robust techniques and highly specific, and we can easily use during the demonstration of the protein-protein interaction using the micro or nanogram of the proteins molecules. As you know that the proteins is very costly, so even we use the micrograms of the proteins in the form of ligands or in the form of analytes. We can easily identify the proteins interactions in vitro conditions. So how SPR is work? This one is completely based on the grasp. When they change the refractive index, these analyte molecules move in the form of waves, and these waves detected in the form of detections area, and these appeared in the form of the graph. This one, lane 1, lane 2, represent the different types of the analyte applied for the interaction studies. Means proteins 1 and protein 2 has the different kinetic parameters at the time of the interactions. So on the basis of the mathematical calculation, we can easily identify if protein 1 is a active interacting partner with the protein 2, or protein 2 is interacting partner of the protein 3. So we can just screen out the whole of the biological molecules through the SPR analysis. Different types of the chips used for the different types of the interactions, like CM5 chips used for the interaction analysis when immobilized by using the functional group of the ligands like amino group, SH group, aldehyde group, hydroxyl group, and carboxyl groups in the interacting partners. All of the process, the ligand immobilization is a key process because all of the ligands molecules are not easily immobilized over the surface. That's why the chip selection is a crucial process for the SPR analysis. Second one is NTA process for the direction of the his tag molecules. As you know that the sum of the proteins, suppose during the purification process, if we tag the molecules with the HIS, with the GST, with the different tagging molecules like biotin, we need to select the different types of the chips for the SPR analysis. Now, SA chips, this one is used for the immobilized biotenated molecules at the time of the protein, peptide, and nucleic acids. So this type of chips are used for the analysis process. Second one is HPA chips that are used for the when we immobilize the hydrophobic molecules over the sensor chips. As you know that the hydrophobic molecules immobilization is very difficult process. The hydrophobic molecules are not easily attached to the surface. That's why we need the specific skilled individual for the immobilization of the hydrophobic residue over the surface. If we are interested to maintain the lipid bilayer integrity at the time of immobilization process, we need to use the L1 chips for the interaction studies. So these different types of the chip are used for the different types of the bioactivity, depends upon the nature of the biomolecules selected and used for the immobilization process. Suppose we have taken the epitope mapping using the SPR analysis. Now, what is the process for the analysis or screening of the epitope mapping using the SPR? Initially, we need to immobilize the antigen over the sensor chip of SPRs. Then allow the binding of the antibody or other partners over the sensor chips. Then real-time monitoring through the instrumental setups and kinetics analysis. We can explain in details the kinetic analysis. Kinetic analysis is different process. We can further discuss in the next slide. Then comparative bindings studies. Now analysis alanine scanning mutagenesis. Suppose ki some of the residue are important for the binding interactions. We can easily generate the mutant by replacement of the one of the amino acids. So different types of the replacement, we can identify which type of the amino acid or residue is important for the interacting process. And then further, we can analyze the all of the sensograms by using the instrumental setups. So this one is different types of the process we can use for the analysis of the protein interactions. And finally, we obtain the value or kinetic parameters in the form of association constant, dissociation constant, and this one is equilibrium constant. Now, we need to observe the data obtained through the SPR analysis. Two of the biomolecules that is belongs to the viral molecules. When we screen the molecules for the interaction studies, we identify there is 5 types of the molecules we can easily identify during the one shot of the reactions. So this one is 1, 2, 3, 4, 5. So this one is 4 proteins. Protein 4 has the lowest interaction activity. This one is molecules 1 that has the higher binding activity on the basis of the sensograms. But this observation is not true. We need to calculate the kinetic parameters, and then we can say the one molecule has the stronger bindings or second molecule has the stronger binding. So on the basis of the sensogram obtained by application of the interacting partners, further we can titrate the 5 different concentration of the molecules. Suppose this one is 1 molecules, we can titrate the different concentration of molecules in the nanograms. Top bottom is 1 nanogram, 2 nanogram, 4 nanograms, and 8 nanograms. So this one is concentration kinetics. When we apply the different concentration in the SPR, we can obtain the kinetic parameters. Kinetic parameters means कि this one is association constant, dissociation constant, and equilibrium constant. Single concentration, we cannot calculate the kinetic parameters of the biomolecules. At least we need to apply than 3 concentration of the biomolecules for the kinetic parameter identifications. Here, this one is monoclonal antibody, and this one is 3 different types of the antigens. I have collected from the paper published in 2023. So how we calculate the kinetic parameter, how we can identify the which molecules has the higher binding affinity. Here we can see all of the biomolecules has binding affinity in the nanograms. These 2 molecules has the binding activity in nanograms. The kinetic, the association constant is 10 to the power 4. The dissociation constant value is 10 to the power minus 3. And the Kd value, equilibrium constant, is 10 to the power minus 8. How Kd value calculated through the instrumental setups? The Kd value equal to the dissociation constant by the association constant, means K off means dissociation value, or K on means the association value. Some of the instrument has the setup to calculate the Kf and K on value. Some of the instrument has set up to calculate the Kd value and Ka value. So on the basis of the association value, we can calculate the highest value is 7.3 in 10 to the power 4. Means these molecules has highest association with the counter partners. On the basis of K value, the second molecules has the lowest association value with the counter partners. On the basis of single parameters, we can easily say this one molecules, first molecules has the higher binding affinity in respect to the second value. But the single parameter is not sufficient to enumerate the binding affinity. Binding affinity mainly calculated in the form by the equilibrium constant. How we calculate? The Kd value equal to K value. So what is the binding affinity? This one is a 10 to the power minus 8. This one is 10 to the minus 8. This one, all of the value is fall into range of the minus 8 value. So lowest Kd value means 1.6 into 10 to the power minus 8. It means that has the highest binding activity. As you know that the Kd equal to affinity constant equal to dissociation constant by the association constant. When dissociation constant value is lower, association constant will be the higher, means that these molecules are strongly bound with the counter partners. Means if we arrange the binding affinity of molecules, this one is 4 molecules in respect to the antigens, we can easily say the first molecules has the strong binding in respect to the second molecules, and this one molecules has the highest binding in respect to this one molecules. Means ki lowest the value of Kd reflect the stronger binding affinity with the counter molecules. So we can calculate the binding affinity either in forms of the bond energy or in form of the equilibrium constant by using the sensogram analysis by the instrumental setup. Now, when we identify the analysis process, that is a kinetic parameters by using the instrumental softwares, Now, next process is targeting of the protein-protein interaction into the drug discovery. We can discuss more details about the protein-protein interactions using the alternative approach. What initially we need to fix the how we can identify the target molecules during the drug discovery using the SPR analysis. So protein-protein interactions can be used for the designing of the drug molecules, but these are the very challenging process because interaction surface are often large, dynamic, and lack of the deep binding pockets. As you know that the different pockets may be opens or may be closed depends upon the environmental situations, depends upon the nature of the proteins, depends upon the residue present over the pockets. The next step, once we identify the protein-protein interactions, we need to characterize the proteins, which part of proteins are signature molecules for the binding interactions. And on the basis of that signature molecules, we can synthesize the peptide molecules and further go for the synthesis of the chemical molecules as a drug for the inhibition of the interactions. Nowadays, the protein-protein interaction is important cellular aspect for the drug designing process. Some of the biomolecules, as examples, Nevotrilex ABT 263, currently in the clinical phase 2, that reported by the Laurent Pollens in 2022. These molecules inhibit the interaction of BCL2 and BCLXL interactions and promote the apoptosis process inside the cells. Second, Q molecules, that is RJ7112, that is in clinical phase 3, Y Yongfang in 2020, that break the P15 and P53 and MDM2 protein interactions and restoring the P53 tumor suppressor activity. So there is huge application of the targeting protein targeting process and drug discovery process. We have used the protein interactions for the drug designing process, maybe in the autoimmune disease disorders, maybe in infectious disease biology, maybe in cancer therapy, maybe in neurodegenerative disease. On the basis of reported literatures, I have collected some of the data like BCL2 inhibitors that already in the clinical phase 2, some of the protein interaction can be used for to minimize the infectious disease like HIV drugs that can be used to inhibit the CCR5 and GP120 interactions and that prevent the entry of virus in intracellular environments. As you know that in case of the neurodegenerative disease, the protein aggregation process is important events that leads to degeneration of the neuron cells, as for example, Alzheimer's. So on the basis of the protein interactions, we can prevent the protein aggregation into the cellular environments and we can minimize the neurodegeneration process. So some of the researchers design the small molecules that break the amyloid fibril formations and that is helpful to prevent the degeneration in the old age people. We can also manage the autoimmune disorders by using the protein-protein interactions. There is report that disrupt the CD80, CD86 with the CD20 interactions to treat the rheumatic arthritis. Similar in case of the cardiovascular disease, we can use the different types of the interaction to block the biomolecular interactions, and all are in the clinical research. Now, for better understanding, we have used the unique examples for the demonstration process. Just like examples, the nitric oxide synthesis inside the cells, there is different type of isoforms of the nitric oxide synthesis present inside the cells, like N neurons present in the neuronal cells, I NOS that is present in the macrophage cells, E NOS that is present into the endothelial cells. So all nitric oxide synthesis that accelerate the process of the nitric oxide generation inside the cells by using L-arginine as a substrate, and that produce the L-citrulline in presence of the cofactors of NADPH. And these nitric oxide is important signaling molecules that accelerate the vasodilations, anti-inflammatory, anti-thrombotic, and neuroprotective response inside the cells. So what happens? The NOS molecules, we need to try the regulatory partner of the NOS molecules. If we identify the regulatory partner of the NOS molecules, we design the a specific inhibitor either against the NOS or against the NOSIP that block the interaction process, and we can control the nitric oxide generation inside the cells. So first process is target identifications. And before target identification, we need to know what is the cellular mechanism of the activity of the proteins inside the cells. Now, suppose ki protein A and protein B interact to each other, that accelerate the process of the oxidation of the L-arginine into the nitric oxides, and that further encodes the biological functions, that is vasodilation, anti-inflammatory, and antithrombotic effect inside the cells. So what happens when the proteins A and B are in separate forms? Protein A has the different types of the domains. Initially, we need to fix what is the buffer situation, what is the environmental situation to open up the pockets. Means ki pocket must be free for the interaction activity. So we need to stabilize the buffer condition, we need to stabilize the pH condition, we need to stabilize the ionic strength required for the opening of this configuration of the proteins. Now, when the proteins A and protein B is open forms, that induce the nitric oxide synthesis inside the cells. Now, when these pockets are in associated form, means when protein B and protein A bind to each other, that inhibit the activation of the nitric oxide synthesis inside the cells, and indirectly it block the all of the cellular event inside the cells, or minimize the cellular events inside the cells through the drug designing process, through blocking the interaction of between the two proteins molecules. So suppose on the basis of the peptide biology, we can synthesize the small molecules on the basis of the homology modeling of the peptides. These small molecules have the ability to bind to protein B or protein A pockets. After the synthesis process, what happens? These proteins molecules inhibit the interaction between the protein A and protein B, and we can control the nitric oxide productions either through the infection stability of the infectious biology or the nitric oxide biology inside the cells. So this one is cellular physiology. What is the significance of the protein interactions? How we control the cellular events by synthesis of the target molecules against the interaction process. Now, second significance is these nitric oxides is a important physiological signaling molecules inside the cells. Again, some of the chemical donors provided in the patients in form of the tablets, and that induce the nitric oxide generation in nitric oxide generation inside the cells. But what is difference? By accelerating the nitric oxide production through blocking the interaction between the two processes, or by providing the chemical inducers that induce the nitric oxide generations, because chemical inducer has the limitation of the shelf life. We can manage the nitric oxide for the milli of seconds, but when we block the interaction, we can manage or balance the nitric oxide up to the minutes inside the cells. So we can accelerate the physiological events according to our need up to the minute. At maximum 3 minutes, we can accelerate the production of nitric oxide inside the cells by using the inhibitors that has ability to block the interaction of the two different proteins. First one is a regulatory protein, and second one is interacting proteins. Now, what is the immunological balance? On the basis of that interactions, if we can induce the transduction mechanism by providing the LPS treatment, the LPS interact with the TLR receptors that induce the nitric oxide activity, and it further activates the L-arginine into nitric oxide synthesis, and these nitric oxides minimize the secretion of IL beta. This IL beta is inflammatory molecules that regulate the inflammation inside the cells. So when ABC nocip is absent, either by generation of the knockout models or synthesis of the small molecules that inhibit the activity of the nocip with the nose, we can manage the IL beta secretion inside the cells, means we can manage the inflammatory reaction inside the cells. Now, second, when nocip is present inside the cells, means that is in active forms. What happens? That increase the IL beta secretion inside the cells, and that again leads to the inflammatory reactions inside the cells. So we can immunologically balance the cells, metabolically balance the cells through the regulation of the nitric oxide generation inside the cells, either through generation of the regulatory knockout models or through secretion of the drug molecules that has ability to minimize the interaction between the two biomolecules. Now, a strategy to target the PPI protein-protein interaction, how sensogram appears after the inhibition process in the SPR analysis. So this one is a chemical library. We can screen out the library, what is the binding affinity with the molecules, either to the analytes or ligands. If this library are suitable to bind with the analytes or with the ligands, that can minimize the positive interaction between the biomolecules. So when in presence of the protein interactions, the sensogram appear in the form of this one. Sorry. So these sensograms reflect the positive interaction between the ligands and analytes. When we incubate the analytes in presence of the chemical library, that molecules bind with the binding pocket of the analytes or binding pockets of the ligands. After the incubation period, then wash out the biomolecules or unbound molecules, and then analyze the interacting process or cellular event in form of the sensograms. So these sensograms reflect there is no interaction or minimize the interaction between the biomolecules. So this one is positive sensograms, means ki positive interaction between the ligands and analyte molecules. This one is negative interactions, means ki inhibition of interactions in absence of the proteins and ligand interactions in the SPR instrument. So this one is a strategy we can use for the identification or targeting the protein molecules for the therapeutic applications. Now, drug development programs that we can develop the drugs. As you know that this one is a time-taking process. Initially, you need to identify the target molecules, then go for the protein synthesis, and then go for the small molecule synthesis, and then screening of the binding affinity by using the SPR molecules. Then go for the in vitro and in vivo studies, that is preclinical studies, and then further go for the clinical trials, phase 1, phase 2, phase 3, and then further go for the approval and really manufacturing and release into the markets. So this one is a time-taking process. If we identify the drug molecules, that approximately take 15 to 20 minutes to launch into the markets. So biophysical technique is important process that is frequently and nowadays frequently used for the screening of the drug molecules or to check the therapeutic potential of the biomolecule inside the cells. Now, second is isothermal titration. Also biophysical techniques that use for the titration of the binding interaction between the ligands and analytes. Ligands and analytes means two biomolecules. Earlier, I have immobilized the ligands molecules over the solid surface. But in isothermal titration colorimeters, both ligands and analyte should be in suspension stage, means ki in random movement inside the solution, and we can monitor the real interaction in the liquid forms. And these affinity molecules we can monitor in terms of release and absorb of the heat energy. Means on the basis of thermodynamics parameters, we can calculate the type of reaction, whether the reaction is exothermic and endothermic, what is the stoichiometric constant between the at the time of the interaction, what will be the enthalpy value of the interacting partners. So this one is instrumental setup for the ITC, isothermal titration colorimeters. We need to fill the references like a blank, means we need to load the buffer used for the protein solubilizations. Now this one is sample cells. Initially we need to fill it with the ligands molecules and then apply the analytes molecule into the sample cells, or initially we need to fill the analytes molecules and then apply the ligands molecule into the liquid suspensions, and then run the instruments. All of the value appear in the form of this one is the heat exchange at the each of the injection lot. So now the sensograms reflect the energy release microcalorie per seconds in terms of the times, and this graph reflect the each and every time of the injection process, the reaction release some amount of energy. Whether this release of energy actually appears through the biomolecular interactions, or this one is a solid suspension of the references, we can tabulate the data by the stoichiometry analysis or graph fitting analysis, Here we can find the graph fitting analysis is perfect according to the calculated value by the instrumental setup. Here we can also calculate calculate the equilibrium constant, and that tabulate the type of the interactions, number of the biomolecules involved at the single point of the interaction, what is the binding energy, what is the kinetic energy of biomolecules through the isothermal titration colorimetric analysis. So this both of the biophysical technique is very important in terms of the calculation of the protein binding analysis, target identifications, and this one is a small molecule synthesis process. Now, second approach is basic computational approach. How we can identify the protein interactions by the strings database or string tool techniques. Now you can know the type of the interaction and inside the cell. Some of the interaction is reversible in nature. Some of the interaction is irreversible in the process. Each of the arrow between the two nodes represent the cellular event. How many cellular event that link with the type of the interaction between the two molecules, either this one molecules or HMG1, HMG2 molecules, or different of the proteins molecules, TLR9 molecules, TLR4 molecules, or CD24 molecules. So this internode represent the physiological event that leads to the interaction of the two molecules. We can also identify the reversible process, means these molecules has the ability to interact with the second molecules, or second molecules has the ability to interact with the first molecules. At the time of the searching by using the string database online tools. Here we have identified HMGB1 proteins as a target molecules, and just try to identify the number of molecules has ability to interact on the basis of computational algorithm. And we found the lot of molecules has ability that directly or indirectly interact with the HMGB1 molecules at the time of the cellular event. We can identify total 9 cellular events can allow the interaction of the HMGB1 and HMGB2 in the human physiology. We can identify there is 2 physiological events that allow the interaction of BECN1 and TLR4 molecules. There is many physiological events that allow the interaction of HMGB1 proteins with the TLR4, with the TLR2, with the TLR9 by a specific counting of the internode that connecting between the HMGB1 to the respective proteins. So this one is overall process for the protein-protein interactions, either by using the computational tools, by using the in vitro approach, by using the in vivo approach. In vivo approach, we can analyze the protein interactions by co-localization process. We can label the one molecules with the fluorescent tag, second molecules with the fluorescent tags, and then analyzed by the confocal microscopy whether the overlapping of the signals appears after the image analysis. So this one is all about the protein interactions and its regulatory natures. As you know that the protein-protein interactions are a critical regulator of the cellular functions and their therapeutic targeting that minimize that has the immense potential and advance in the structural biology and computational modeling and drug discovery that may continue and expand the landscapes of the protein-protein interaction in the therapies and that minimize the scientific gap present till now. Thank you. Now I am open for the questions.
Thank you, sir, for that enlightening talk on protein-protein interaction. Do we have any questions? So I think, sir, there are no questions. Everybody has understood everything.
Okay. Thank you.
Okay. Thank you once again, sir, for your informative lecture today and your time.
Thank you. Thank you. Thank you. Bye. Okay.
So this basically brings us to the end of the fourth day lecture. So tomorrow is the last day, and all the lined up speakers and their details will be shared in the group. Okay. Thank you and have a good day.
So now we move.